Hello class. Today we are going to see the chapter DNA damage, detection and repair system. Which topics to be included in this chapter? Firstly, the introduction. Next, DNA damage and its types of DNA repair, pyrimidine dimer formation and its repair. Introduction. As we know, DNA is highly stable and a complex organic molecule. It is very susceptible under normal cellular conditions. The DNA, when is exposed to any radiation or any chemical component or chemical reaction, the damage is occurred and the bonds get broken down. The DNA damage is occur due to alteration in the chemical structure of a DNA, breakage of bonds, missing of bases from the backbone of a DNA. How it is caused, or which are the factors which cause the DNA damage? Radiation, chemical mutagen, heat, enzymatic errors, spontaneous decay constantly damage the DNA. This process is occurring in normal cells also. When the metabolic reactions are carried out, some enzymatic errors are coming in contact, so it is cause damage to the DNA. There are two sources that causes the damage. The exogenous sources includes the external agents like radiation. These radiations are UV rays or X-rays. Then the next source is endogenous sources, which includes the byproducts of metabolic process like reactive oxygen species and some replication errors also. Now, what are the types of damage? Single base alteration, two base alteration, cross linkages and chain break. Firstly, we see the single base alteration. What is meant by alteration? It means that alter the structure or bases within a DNA molecule. It is an alteration in the single bases within a DNA which differs from the normal form of a DNA. This single base alteration includes depurination, deamination, alkylation and some replication errors. What is happening in depurination? It is a chemical reaction in which the beta and glycosidic bond is break within the purine nucleotide. It creates a purinic site which is occur due to the radiation alkylating agents or free radicals. What is deamination? It is a removal of amino group from the molecules. The alkyl group is get transferred from one molecule to another molecule, which is the alkylation. In the process of replication, some base analogues are get incorporated into the normal DNA. So it causes the damage. Here we see the depurination and deamination. When the reactive oxygen species is reacted with the structure, the guanine undergoes the reaction and the depurinated sugar is get formed. In deamination, the amino group is removed and the uracil is get attached to the DNA strand. In normal DNA structure, uracil is not present, so it is a damaged DNA. The next one is 2-base alteration. In this method, the 2 bases were get altered and form a dimer which changes the structure of a DNA. When DNA is get exposed into the UV radiation, it forms the lesion from thymine or cytosine. UV radiation causes the formation of covalent linkage between the bases. The lesion alters the structure of a DNA and it inhibits polymerase activity, which results in the stopping the process of replication. Then the third one is cross-linkage. It is occurred due to exposure of UV light, UV rays, free radicals like hydrogen peroxide. Hydrogen peroxide, it forms a complex with the protein and it causes the linkage between the DNA and the protein. Then the next one is chain break. When DNA is get exposed to the radiation, it causes the base alteration or analog alteration, which inhibits the polymerase activity. When the polymerase activity is get stopped, the replication get collapse. Due to this, the DNA get broken down. Now we are going to see the types of DNA repair. How the single base alteration or cross linkage is causing and how we can repair that DNA sequence. There are 6 types: direct repair, base excision repair, nucleotide excision repair, mismatch repair, recombinational repair, and source repair. How this system works, let us see. First one is direct repair. In this, we are taking the example of pyrimidine dimer formation. These are the UV rays, which one? UVA, UVB and UVC. These are the types of UV radiation, which having the wavelength ranges 300 to 400 nanometer, 280 to 320 nanometer, and UVC falls less than 280 nanometer. UVB is mainly cause the damage to the DNA. It breaks the bond between the nitrogenous bases and form the dimer. It affects the expression of multiple genes and result in the production of pyrimidine dimer formation. In this structure, here the 2 adjacent thymine molecules form a dimer due to breakage of bonds Then the next one is base excision repair. What is meant by excision repair? It means that the damaged portion of a DNA is get removed and the gaps are filled or patched using the complementarity with the remaining strand. In base excision repair, the bases can remove directly by radiation or hydrolysis or attack of free oxygen radical or the enzymes. The DNA glycosylase enzyme sense the damaged bases and remove them. If we consider the example uracil DNA glycosylase, it is a product of UNG gene which recognize the uracil within a DNA and it cleaves the sugar and base glycosylic bond. It creates the AP site, which is a purinic site, which means it lacks the purine or pyrimidine at that site. Enzyme AP endonuclease sense the minor distortion of a DNA double helix and initiate the excision of a single AP nucleotide in the process, which is known as base excision repair. This AP endonuclease create a nix at 5' site and form base-free AP site. The DNA polymerase then insert the nucleotide at AP site. Then exonuclease lyase or phosphodiesterase enzyme then remove the base-free nucleotides. The DNA ligase then encloses the nicks which are getting formed. In this diagram, it is a uracil which is incorporated in the DNA sequence. In the normal DNA, uracil is not present. The guanine is paired with the cytosine. When the uracil DNA glycosylase enzyme is working on that site, it removes the uracil. Then the AP endonuclease cleave that site. Okay. Then the DNA polymerase adds the nucleotide at AP site, and then the DNA ligase is filling the gaps between the sequence. Then the next one is nucleotide repair. Like we see in the base excision repair, here the nucleotides are getting incorporated at a damaged site, or the damaged site is removed and the nucleotide sequences get added. This repair system is initiated by some enzymes. These enzymes recognize the damage in the DNA backbone and replace the short sequence of nucleotide. In base excision repair, we see the bases are getting added to the sequence. In nucleotide repair, the short sequence of nucleotides get incorporated in place of the damaged DNA. If we consider the example of E. coli, it consists of 6 enzymes which excites short stretch of a DNA which contain the thymine dimer. The 2 copies of a protein product of UVRA gene combine with the 1 copy of product UVRB gene, which forms the complex that move along the DNA, and it looks for the DNA damage. When the complex find the damage, such as the thymine dimer, it moderate to large distortion of a DNA double helix. The UVRA second dimer is get dissociated and it leaves the UVRB subunit. Due to this, the UVRB subunit is alone, so it causes the bend at the DNA and attracts the protein product of UVRC, which is a product of UVRC gene. Then the UVRB subunit is first nicks the DNA. 4 to 5 nucleotides at 3' site of the lesion is get added. Then UVRC subunit nicks the DNA and add 8 nucleotide at 5' site of the lesion. The enzyme helicase 2, which is a product of UVRD gene, removes the 12 to 13 bases oligonucleotide and UVRC also. Then the DNA polymerase first fill the gap and UVRB and DNA ligase closes the remaining nicks. It is a complex of UVR-A2 and UVR-B which located at the lesion. Okay, then UVR-A2 get dissociates and UVR-C is get attached at the lesion. Then UVR-B and then UVR-C nicks the DNA. Then helicase unwinds the oligonucleotide with the dimer. Then DNA polymerase and DNA ligase adds the nucleotide and the gaps were filled. The next one is mismatch repair. What is happening in this repair mechanism? In the replication, the DNA polymerase replicates the DNA. Some errors are produced due to proofreading activity of polymerase. At that time, some errors are made which does not get corrected. If the template G is paired with the T rather than C of a progeny strand, the GT base pair does not fit correctly in the DNA duplex. In this system, this error is get recognized. Again, we considering the example of E. coli, which having the specified genes MUT-H, MUT-L, MUT-S and MUT-U genes. They are responsible for incorrect basis removal by the excision repair system. This mute means these are the mutators, means they causes the mutation at a gene at high level like this spontaneous mutation. In the mismatch repair system, the enzyme initiates the removal of incorrect bases by nicking the DNA strands on one side of mismatch. How the mismatch repair system recognizes the progeny rather than the template base. The copies of same protein get find out the MUT-L gene is form the homodimer and binds, and they together find the methylation signals. It's also activate the endonuclease MUT-H, which then nicks the unmethylated strand, which consists of a sequence 3' CTAG 5' recognition site. It is recognized by MUT-H. At the recognition site, MUT-S and MUT-L loads the helicase, which is MUT-U, which unwinds the nicked strands. Any one of the 4 different exonucleases then attacked the unwound oligonucleotide. DNA polymerase 3 then repaired the gaps and DNA ligase sealed it. This sequence of events highlights the common theme in the DNA repair. Once the lesion is found, the damaged DNA has some protein bound it until the repair is not get finished. It is the diagrammatic representation of mismatch repair. Here we see the G is attached to the thymine. When MUT-L binds, the methylation signal is get produced. Then the MUT-H endonuclease form a nick at GATC site. Then the MUT U binds and unwinds the nicked progeny strand. Then the exonuclease digests the sequence. Then the polymerase and ligase add the nucleotides and the DNA ligase fills the gap within the nicks. The next repair system is recombinational repair. In the replication, when the damaged DNA is seem to be repaired, the new strand or the copy which is getting created newly or synthesized newly, it helps to recover the damaged DNA when the polymerase leaving the gaps. Then the new copy of a strand is used for the damaged DNA to be repaired. At the replication fork, it creates the 2 DNA duplex. The undamaged copy of a region with the lesion exists on other daughter duplex. The group of enzyme specific by the REC-A locus having the importance that it repairs the gaps. The repair takes place at a gap which is created due to the failure of DNA replication. This process is also known as post-replicative repair, and it shares the many enzymes which are helpful in the recombination. It is a REC-A dependent post-replicative DNA repair. In this, the thymine dimer is recognized by the enzymes, and these are the newly formed strands. Then the last repair system is source repair system. When any cell is get exposed to the excessive quantity of UV light or other mutagen or other chemical agent, it damages the DNA. Or when the replication is inhibited, the gaps are created in the DNA strands. The other genes include REC-A, UVR-A, UVR-B, and UVR-D. These are the genes that inhibit the cell division. Then SULA and SULB and several others. Each of these genes are Each of these genes has consent sequence in the promoter, which is called as source box. Source box consists of 5' CTGX CAG 3', in which the X is referred for any basis. The LEX-A protein normally binds at source box, which limits the transcription of that gene. When a single-stranded DNA activates the REC-A protein, it interacts with the LEX-A protein and triggers the autocatalytic property of LEX-A. Then the transcription follows from all the genes having source boxes. The two inhibitor of the cell division, the product of SULA and SULB genes, which increase the amount of time the cell has repair the damage before the next round of replication. It is schematic representation of the source repair system. It is a source box. Then UVRA, UVRB, UVRD, SUL, SUB, SULB and other enzymes which are required or proteins which binds to the source box. Okay, then it activates the REC-A protein which causes the catalysis of LEX-A, which eliminates the repression of all the locus present at that site, and repression is occurred, then are transcribed and translated.